next presenter, Jacob Lusk, is going to wow us with his butterflies.
Hi, as he said, my name is Jacob Lusk. I'm a rising sophomore here at the university studying ecology, evolution, and biodiversity. Now, if you couldn't tell from the mouthful of a title and the very on-the-nose t-shirt, I'm here to talk to you about monarch butterflies. Now, most people have something of a familiarity with this very famous bug and the incredible migration it takes every year, from your backyard to Mexico and back again. But what few people are familiar with is another very interesting behavior that monarchs exhibit, namely that of self-medication. Not with drugs, but with plants. You see, many populations of monarch are afflicted with a nasty protozoan parasite known as OE. It's that little microscopic guy in the red circle. When a caterpillar comes down with OE, it often doesn't make it past the cocoon stage. And if it does, the adult struggles to survive the long migration. However, monarchs are not defenseless. They do have a way to combat OE. See, caterpillars feed exclusively on a type of plant known as milkweed. Milkweed gets its name from a goopy, milky poison that oozes out of their leaves when damaged. An expecting mother will choose to lay her eggs on specific species of milkweed depending on toxicity. Here, I define toxicity as the presence of a steroid known as cardenolides, which is found in that goopy, milky stuff. If the mother has the parasite, she'll choose the more toxic milkweed. If she doesn't have the parasite, she'll choose the less toxic milkweed. The idea being that this toxicity helps the caterpillar fight off the infection. My project consisted of a controlled experiment that looked at precisely how this toxicity helps a caterpillar fight off the infection on a molecular and genetic level. This is also what I researched last semester when I presented on at the spring symposium with my partner Andrew Campbell and this eyesore of a poster. And back then we had a hypothesis that the caterpillars that ate the more toxic plant would show a greater expression of genes related to immune defense. What we found was a little different. Firstly, we saw that expression of immune genes did increase for all caterpillars, regardless of diet, as they got older and transitioned through the early stages of development. What we also saw, however, was that those caterpillars that ate the more toxic plant actually had a smaller increase in immune expression. Of course, this is exactly the opposite of what we had expected. A little confusing at first, but once we got to thinking about it, it did make some amount of sense. The bugs we tested here were uninfected. They didn't have OE. So for an otherwise healthy individual to be eating up this goopy toxin, it's probably not a great idea. These preliminary findings had us inspired to revamp the project this summer. We redesigned the experiment, still with 3 developmental stages known as instars, but for each instar, we infected some individuals and left others uninfected. Among these subgroups, we had some feed on the more toxic milkweed and some feed on the less toxic milkweed. Now, what did this actually consist of? First and foremost, we had to collect the eggs. Once the caterpillars hatched, we placed these newborn larvae on little pieces of milkweed and either infected it or left it uninfected. Once they finished that, we let them feed a little longer, then took a few individuals and flash froze them with liquid nitrogen to preserve their RNA. And we repeated that, all those steps again and again until we had samples from each instar. However, there are a few more steps, but that isn't as far as we got. We still have to extract the RNA, turn that into DNA, run tests on that DNA, and then compare all those results. Now, I'm leaving in a few days, so I'm not going to be able to see this project through, but I'm passing the buck off to my colleague, Kathleen Young. Now, once Kathleen finally finishes up these last steps, what do we expect to find? Well, our hypothesis this time around is in part the same as it was before. For the caterpillars that eat the more toxic milkweed, we expect to see a greater increase in expression of immune genes, provided that they are infected. For the caterpillars that we left uninfected, like our last experiment, we expect to see the same trend we saw last time too, with the uninfected caterpillars consuming the more toxic milkweed and having a smaller increase To put it more simply, if the bug's infected and eats the more toxic plant, it'll have a greater increase in expression. If it's uninfected and eats the more toxic plant, it'll have a smaller increase. Now, I just talked a lot of science. You might be wondering, where was I in all that science? What was my role in this project? Well, it was pretty much all over it. My partner, Andrew, and I were involved in every aspect of the experiment. We caught the butterflies ourselves, we cultivated the milkweed, we raised the caterpillars and hand-fed all the adults. We performed all the experimental procedures to a point in the lab, in addition to designing each step of the experiment ourselves. You might also be wondering, monarchs and parasites, who cares? Why study this seemingly niche aspect of monarch biology? Well, the reasons are threefold. For one, as you can see, it's pretty cool. I had a lot of fun. It was an incredible privilege to study these fascinating creatures. For two, the information we find here will hopefully be broadly generalizable to the up-and-coming field of behavior immunology. Behavior immunology studies how an animal's behavior affects their ability to defend against pathogens, infections, their health and their immune defense. This is most commonly seen in humans, both by use of medicine, practice of exercise, or lack thereof. But it can also be seen across the animal kingdom, from chimps to birds to monarchs. Thirdly, and most importantly, is that while we all know and love monarch butterflies, they are disappearing. Now, OE is not the only cause of this decline. Climate change and habitat loss have a lot to do with it. But it is a contributing factor. If we can better understand the relationship between this parasite and this insect, we can better protect monarchs in our conservation efforts. Lastly, I'd like to thank all the people that helped make this project and this presentation possible. It was definitely a team effort. And if you have any questions, I'll take them now. Thanks for listening. Yeah.
What made you first interested in this kind of a project, so to speak?
Well, I started last October, and really, it's just the butterflies. Like, you gotta love butterflies. They're pretty cool. Like I mentioned, I'm studying evolutionary biology, so I was interested in working with something related to animals and to genetics to see how it felt. Especially the wet lab experience that I was gonna get seemed attractive and ended up being a good experience. Any other questions?
What should we know as everyday citizens about butterflies and caterpillars?
Say that again?
What should we know as everyday citizens about caterpillars and butterflies? What's most important?
That's an interesting question. Well, they're hardier than you think. If you touch it, it will not die. That being said, don't go around grabbing butterflies, but I hear a lot of people go like, oh, you touch them? Aren't the oils going to ruin the wings? And they probably will, but butterflies are designed to go across this whole migration. They're pretty hardy, so don't be afraid of these delicate little creatures. And also, if you want to track monarchs, it's a good idea to plant different species of milkweed, which is kind of the project, because they might not want the milkweed you have in your garden. So if you want to track monarchs, plant like a bunch of different kinds. Yeah.
How did you keep the butterflies, how did you keep them separate or know which ones were which?
Right, so we have multiple outdoor enclosures at the botanical gardens, and inside those enclosures we have separate cages, and we actually label the butterflies with a Sharpie. So that's how we keep track of it. We label them and we create identification cards so we can keep track of different genetic lineages and different geographic origins, things like that. Yeah?
Do the butterflies self-medicate when they're infected with anything else besides the specific
I can't say that I know the specific answer, but in your best guess, I imagine they would, but I'm pretty much just an OE expert at this point. Yeah?
What's the best time to see butterflies in Michigan?
The best time to see butterflies in Michigan is just this past month and this month coming. They started arriving back from Mexico about not that long ago, maybe 3 weeks ago, month. So they're just arriving, they'll be here a little longer, and then they'll start leaving again in the next month or two. All right, thank you very much.